So good evening, folks. Thanks so much for coming tonight to hear me talk about my work on the Maine Coastal Islands National Wildlife Refuge. And tonight I'm going to be talking about work that I've been doing over the last 7 or so years, looking at decadal and seasonal patterns in seabird foraging ecology across the Gulf of Maine. So my name is Dr. Michelle Staudinger. I'm an ecologist with the U.S. Geological Survey. I'm the science coordinator for the Department of the Interior Northeast Climate Adaptation Science Center, and I'm faculty at the University of Massachusetts Amherst. So in my roles with USGS and at UMass, I study climate change and its impacts on coastal and marine biodiversity. So for context, I wanted to start out with a little bit of background on what we know about rising temperatures over recent decades, both globally and in our region. On a global scale, we know that temperatures have risen in line with carbon dioxide concentrations, with the majority of increases occurring since the 1980s. We've already experienced about 2 degrees Fahrenheit of warming since the early 1900s, as shown in this graph here. And this trend is reflected in both air and sea surface temperatures. When we look at this global map of sea surface temperature anomalies, we see that the Gulf of Maine really lights up as a hotspot of warming. These data that I'm showing in this map here are from the year 2021, which was the 4th warmest year on record for the United States and the 6th warmest year globally. And when we look at the sea surface temperatures in the Gulf of Maine, we see that the observed changes over the last several decades are at a rate that is 2 to 3 times faster than the global average. And we see these strong warming trends during all seasons, with some of the highest rates over the ocean in the summer. This is really important because a number of species migrate to the Gulf of Maine to take advantage of the high productivity during the warmer months and use this for their seasonal foraging and breeding grounds. Climate change induced warming trends impact all life on Earth and also in the Gulf of Maine. And we have extensive and mounting evidence that biodiversity is responding to climate change through widespread changes in distribution, range, production, and phenology of organisms. And phenology is the topic that I'm going to be focusing on today. So what is phenology? Well, we define phenology as the timing of critical reoccurring life events that occur at a particular date and a particular place in a species' life history. This can manifest as migration, breeding, and developmental schedules or growth rates. Phenology has been most commonly discussed in terms of plants and bud burst, spring bud burst, and the flowering, the bursting of flowers as the temperatures warm in the warmer months. But as you can see here, it can also affect some of the other life history events in other organisms. Phenology can also be described as the timing of trophically linked foraging relationships, which brings me to the main focus of my research program that I've been doing in the Maine Coastal Islands National Wildlife Refuge with seabird populations, as I mentioned, over the last 5 to 7 years. These images here show examples of several of the seabird species that I've been collecting foraging ecology data on in the region. And all the prey in these images are species of forage fish that are typical in their long-term historical diets. But recent assessments of climate risk for seabirds in the Gulf of Maine and throughout the Northeast US have raised concerns that seabirds may be highly vulnerable to climate-induced reductions in prey, especially during the sensitive time when they come to the Gulf of Maine region to breed and raise their chicks. And there's one fish in particular that has caused a lot of concern, and that is the butterfish. Observers in the Maine Coastal Islands have observed this warm water associated fish being provisioned to seabird chicks during poor survival years. And it is hypothesized that the birds are feeding their chicks butterfish because they can't find their preferred prey. Butterfish are problematic for a number of reasons. They are deep-bodied and they're very difficult to swallow compared to some of the more narrow and elongate prey that terns and other seabird species typically prefer. They're also thought to be less nutritious, having lower lipids and fatty acids than some of the more typical prey that we've seen historically in seabird diets. The ecological implications of the shifting availability of prey are thought of as trophic mismatches or asynchronies. And these asynchronies or mismatches can affect individual fitness, growth, and survival. But proving that a phenological mismatch is actually happening is actually really difficult to measure. Therefore, there are really relatively few definitive examples that we can point to. I always like to show this image when I discuss phenological mismatches, because this is a real exception to the rule of sort of a difficult example of mismatches. In this example of, we can really clearly see that the snowshoe hare's coat color is mismatched with its surroundings. And we know from field studies that this makes it much more vulnerable to predation than a camouflaged hare. And this mismatch is happening more and more during recent warm years. And so determining whether nesting seabirds are becoming temporally mismatched with their prey is a primary underlying objective for the research that I'm going to talk about today, which is to determine if the shifts in the temporal abundance and distribution of prey are causing these phenological mismatches in the Gulf of Maine system. And if these mismatches are happening, what are the consequences for the multiple species of terns that nest on the Maine Coastal Islands National Wildlife Refuge? A quick primer on tern foraging ecology. Like other seabirds, terns are highly mobile and can fly great distances. Some of these species, such as the Arctic tern, migrate as far as the Southern Ocean, earning them the award for making the longest migration of any animal on Earth. But during the nesting season, terns and other seabirds are place-based or central-based foragers, and they generally hunt for food within a 12 to 25 kilometer radius around their nesting sites. Now, the further they have to fly to find food, the fewer trips they can make back to their colonies and feed their chicks, and the more energy they have to expend. So this can affect their fitness and their nesting success. These smaller seabirds are also restricted in that they can only access food in the top few centimeters of the just below the water surface. Terns forage by plunge diving, and this is in contrast to some of our larger diving seabirds like our alcids, the Atlantic puffin and razorbills, for example, which can hunt and access food several meters beneath the surface to find larger prey fish. Terns also differ in their foraging habits compared to their larger alcid cousins, as they typically only carry a single prey item at a time back to their chicks. So in these two pictures, the one on the left shows a tern with just a single prey item. It's actually an Atlantic herring. And on the right, we have a razorbill, which has a beak full of multiple herring. So even though the two pictures show the same prey, in these two different seabirds' bills, the fish that the tern has caught is only about two inches in length, while the razorbills are feeding on adult herring, which are probably about 8 to 10 inches in length. My lab has largely focused on 4 species of terns that nest across the Maine coastal islands. This includes common terns, Arctic terns, roseate terns, and least terns. We've worked to quantify the long-term trends in tern-chick foraging ecology across 7 islands, which include Stratton Island, Outer Green Island, Jenny Island, Eastern Egg Rock, Matinicus Rock, Pond Island, and Seal Island National Wildlife Refuge. And we've worked to amass over 30 years of data, which we have used to characterize these long-term trends in diets, and looking for patterns over decadal, annual, and seasonal scales. Some of the more recently collected data were already in a database, which is organized and maintained by National Audubon and US Fish and Wildlife Service. But some of the older data that was collected prior to the year 2000 had to be digitized before we could integrate it into a full comprehensive data set to then analyze it for these long-term trends. So here's an example of how these data are collected. As I mentioned, the Maine Coastal Islands has a long-term monitoring program, which is overseen by the National Audubon Seabird Institute and U.S. Fish and Wildlife Service, who have been collecting data on chick provisioning survival since the mid-1980s. This video shows an example of how these data are collected. So we have an observer in the bird blind, which is looking down on a given area, which has multiple nests of chicks, which they observe at a given time. And from this information, we can see what prey was delivered, how big the prey was, which chick the prey was being delivered to, and we can get information on trip times in between when the adult left, went out and hunted for food and returned. And we can also get information on, I think if I didn't already say, how big the prey was. And so this prey that was just delivered here in this video is actually a sand lance. And you can see that this chick is able to just slurp down that sand lance really quickly. Again, as I mentioned earlier, that butterfish has this really deep body, is very difficult to swallow, while these sand lance are very easy to sort of slurp down, kind of like spaghetti. All right, so when we looked at over 30 years of data collected across the islands, here's what we found. We found that between 1988 and 2018, that over 70% of the collective tern chick diets across the 4 species of Arctic common, roseate, and least were comprised of 3 main prey groups: hake, which is mostly white hake, but also contains percentages of offshore and silver hakes, 2 species of sand lance, an inshore and an offshore species, and a herring group, which is mostly Atlantic herring, but also contains unknown amounts of river herring species, which include alewife and blueback herring. And then the pink in this figure sort of encompasses all other species of fish. So this high specialization on a few prey species is a known factor to increase the species' vulnerability to climate change and other environmental factors. This next plot shows the relative chick diet diversity across the 4 species, which is derived from metrics quantifying the total unique prey species in each tern species diet. You can see that Arctic terns and common terns, which are on the left-hand side of this plot, have more diverse diets. This is in part because they're able to provision invertebrates to their chicks, which includes a mixture of both marine invertebrates like amphipods and small crustaceans, but they'll also hawk terrestrial insects out of the air over the islands and feed those to their chicks on occasion. Roseate terns and least terns have less diverse diets. Both of these species are primarily piscivorous. And across their range, roseate terns are actually considered to be sand lance specialists. When we compile data from colonies that went as far as the mid-Atlantic all the way up through the Gulf of Maine and into Canada, we found that roseate terns diets range between 30 to 100% of their diets containing sand lance, one of the 2 species of sand lance. The least tern diets may be less diverse compared to common and Arctic terns because least terns only nest and provision their chicks on Stratton Island, which could lead to just a lower overall diversity compared to where we have data across multiple islands. And for both least terns and roseate terns, the data in this study are some of the most comprehensive that have been collected to date across the region, and one of the first studies of least tern diets in the Gulf of Maine. So although the terns diets are highly focused on these 3 prey groups, the hake, the sand lance, and the herring, there were some really interesting results that indicate that they may have the capacity to utilize novel prey conditions that may be brought about by warmer years under climate change. So during 1992 on Eastern Egg Rock and on Jenny Island as well, we saw that roseate terns and common terns provisioned unusually high amounts of pollock to their chicks. Pollock are a species of gatid similar to hakes, Atlantic cod, and haddock. And we think that the pollock may have experienced a population explosion during that year, which made them just overwhelmingly abundant in these locations. This is not on this does happen on occasion. I have a colleague who has collected data at some of the more southern colonies, tern colonies off of the coasts of Massachusetts and New York. He was doing work in the 1970s and noted that although common terns and roseate terns at those more southern colonies eat almost exclusively sand lance in those places, he remembered years in the early 1970s where they appeared to shift their diets away from sand lance, and he called these instances the mackerel year, the round herring year, and the anchovy year. So these sort of anomalous events do happen, but we expect that they're going to continue to happen more often under climate change, and that some of the more typical prey that the birds rely on will be replaced by warm water species that are moving into the Gulf of Maine in response to climate change. So this next series of plots shows examples of the decadal trends in common tern diets. And the main point that I want you to take away from this is that the importance of each of the major prey items varied quite a bit over time and location. This is important because it indicates that at the different colonies, the local areas may experience different conditions and different assemblages of preys even in the same year. So changes in prey populations don't seem to be affecting the tern colonies uniformly across the region. So for example, if we look at Staten Island, we follow the sort of tan line, which is representative of sand lance population, we see that sand lance were really important in their diet, around 40% from the late 1980s through the early 2000s. And then we saw a drop in importance in the early 2000s, and then a rebound in the more recent decade. Now, if we look at that in comparison to Eastern egg rock, we see that sand lance are sort of lower overall frequency in the diets across the entire time series, with some sort of minor ups and downs. And somewhat similar, Jenny shows lower abundances overall in sand lance with a slight rise in the frequency of occurrence in the last part of the time series. Contrast that with herring, we see sort of some ups and downs at Staten Island. This is depicted in the green with an increase and then another decline in around the year 2010. Whereas in Eastern Egg Rock and Jenny Island, we see that there were some sort of different peaks in abundance in the diets earlier in the time series, but then both show increases in herring in more recent years. In general, across the series, we see sort of low levels of this other fish category in pink and the butterfish category in orange, although we do see some more higher peaks, for example, in at Eastern Egg Rock around 2010 and also again in Jenny Island. And I'll mention that the colors that are associated with the different prey fish will carry through the rest of the next several slides. So next, we evaluated long-term trends in the seasonal chick foraging ecology. And so here I'm showing some plots of the within season trends of a couple of the key prey fish species. And in this plot on the left, what I've done is I've matched up what we use here is year day, or the day of the year, is that we see the frequency of occurrence of the different prey in the tern chick diet, and then we've lined up those year days with the actual dates. So year day 170 matches up to around mid-June, which is when we see the majority of eggs laid. Early July, we start to see chicks hatching around year day 192 or so, and then towards year day 215, July August, where we start to see the tern chicks start to fledge. And so when we look at the within season phenology, we see that our cold water associated prey fish are more important earlier in the chick rearing period. So they're much more abundant. Hake is much more abundant in mid to late June, early July, sand lance as well. If we look at herring, though, we start to see that the pattern is quite different, and we see that herring are much more important later in the chick rearing period, increasing towards late July, early August. And we also see that trend for our warm water butterfish and this other fish category. And I'll come back to this in just a moment. So the next thing that we evaluated was how changes in sea surface temperature affected the occurrence of these major prey groups in the chick diets. And each line in this plot represents one of the 7 main coastal islands that we had diet data from. And in this first plot, we see a strong negative relationship between the cold water associated hake and increasing sea surface temperatures. Interestingly, and contrary to expectations, we did not see an increase in butterfish with warming sea surface temperatures, except at one site, and that's Matinicus Rock, whereas the rest of the islands actually show a decreasing trend. Another interesting result was that with increasing sea surface temperatures, we saw an increase in the other fish and overall dietary diversity category. And so we've hypothesized that this may be the result of the terns having to supplement their diets with alternative prey options as their more preferred cold water associated prey becomes less abundant or available. And this may correspond also to that sort of late season phenology that we saw in the previous set of plots. Okay, so what did we learn from evaluating these long-term data on chick diets? Well, first we learned that the 4 species of terns have relied heavily on 3 prey groups: our hake complex, our herring complex, and the 2 species of sand lance during the last 3 decades. But the importance of each of these groups varies in the tern diet by location across the time series. We also see strong seasonality in prey occurrence in the chick diets that suggests that cold water prey are more important earlier in the chick rearing period, and warmer water prey are more important later in the chick rearing period. And finally, we see a strong influence of warming sea surface temperatures on prey occurrence in tern chick diets, which suggests that these cold water species may be becoming less available during warmer years. So going back to my initial question of whether or not terns are becoming temporally mismatched with their prey, we still don't really know that definitively yet. And this is because although we know that what the birds are provisioning to their chicks, we don't have good data on what the distribution and occurrence of the fish are in the local inshore environments around the islands. Therefore, we don't know if the occurrence of prey in tern diets reflects searching and hunting behavior by the birds or the availability of prey in the environment. So this video here is actually taken off of Stellwagen Bank, which is showing a feeding frenzy on sand lance. And I think there's actually multiple species that are foraging on the sand lance all at once here. So we've got the birds foraging on the top, and there's probably some whales that are actually bubble feeding and pushing the sand lance up to the top of the water surface so that the birds can access them. My colleagues who work on Stellwagen Bank have been working very hard to try and collect distribution and abundance data of sand lance in that system, but we really don't have a great sense of where sand lance are. So prey distribution can be highly variable, both in space and time. And this figure shows data that we got from the Massachusetts Department of Marine Fisheries. This is from their spring trawling survey, which they conduct during the month of approximately May and June each year. And oops, sorry. And what I wanted you to see in this figure was how on an annual basis, the number of sand lance that they caught in this survey really shifts around spatially, but it also kind of blinks in and out in terms of the maximum abundance, which is this darker color here. And you can see that it's just highly variable. And this is really typical of many of our forage fish species, which have these boom and bust population dynamics, which make it really difficult to predict their occurrence across the region, especially in places where we're not sampling very often. Another limitation to understanding fish prey abundance and distribution is that we don't have great data on the early life stages of fish populations, which include those larval and juvenile fishes, which are tern species that I've been talking about so far today, rely really heavily on. Sand lance in particular, again, comes up as a really data poor species because they have a winter early life history phenology. So they, their eggs are spawned in the late fall, early winter, and then the eggs hatch and the larvae grow throughout the winter, which is not a time of year when we're out on boats sampling the plankton in a comprehensive way due to just the nature of going out at sea in the middle of winter. Sand lance are also really difficult to get a handle on their populations because they have this really cool and unique behavior of burrowing in the sediment. This allows them to hide from predators by diving. You can see these fish in this video diving in and out of the sediment. So it allows them to hide from predators, but it also allows them to evade our nets. So our typical survey gear that is very good at catching a lot of other fish don't actually catch sand lance very well. And then finally, sand lance are an unmanaged forage fish, which means that we don't collect regular information on their population trends as we do of commercially harvested species like herring and hake. In contrast, Atlantic herring are a managed stock, fishery stock, and we do have information on their population recruitment and abundance. And in fact, when we look at the overall amount of herring and tern diets and look at it relative to herring recruitment into the fishery, we see a really strong relationship. So in this figure, we have herring recruitment in the billions on the x-axis, and then the percentage of herring in the tern chick diet across those 7 islands using those data that I mentioned earlier. And you could see that there's just this really strong positive relationship. So when we have higher recruitment, we have higher amounts of herring. We see higher amounts of herring in the tern diets. So the question still remains as to whether terns and other seabirds can sustain changes in prey abundance due to the shifts in phenology in the Gulf of Maine brought about by climate change. Global evaluations of seabird populations have suggested that seabirds are not shifting their phenology in line with the observed trends in global temperature. But we know that many fish species are shifting their distribution spatially north and to deeper waters as they seek thermal optimal conditions. Less is understood how shifts in range and timing of these species are affecting the seabirds in the local inshore environments around the Maine coastal islands. So how can we get better data on this? How can we assess these data gaps? Well, first, we need to continue to monitor seabird populations to get high quality data on what they're consuming and look for evidence that they are finding and eating novel prey as climate change changes the assemblages of fish that are occurring in the Gulf of Maine. The long-term data sets collected at the Maine coastal islands are really unique and critical to understanding climate-induced shifts in populations. And I can't stress enough how valuable those continued collections are. At some of the more southern colonies in the Northeast US that are located off of Long Island Sound and in Buzzards Bay up south of Massachusetts and Cape Cod, we're working to reproduce data collections to match the quality and extent of the data in the Gulf of Maine. But we have to look into more remote sensing methods like using cameras to collect the same kind of data because we lack the ability to access the island have lived to collect those really great data that are collected on Maine coastal islands. There's also staff capacity issues at those southern colonies. So it's much more difficult for us to recreate those similar data. So this video here that I'm showing is some data that was collected from a camera trap, and we're basically trying to work out the methodology so that we can try to place these cameras out on these islands and not necessarily have to have them stacked at the same capacity, and maybe we can start to collect similar data. Next, we need to collect better data in the inshore environment on fish populations to resolve that question as to whether diets are reflective of seabird behavior or the abundance in the environment of those prey. We can do this through a number of methods. Seining and plankton surveys are common methods to collect information on fish distribution. And some of these data do exist, but they're located in different places and need to be synthesized into similar formats so we can use them to reconstruct a better picture of what's happening in the Gulf of Maine. And this is really in contrast to some of the state and federal surveys that sample further offshore, and are collected on a regular basis. Those federal and state surveys, they really reflect the trends that are going more on the continental shelf and shelf edge, whereas we have just less consolidated and comprehensive data in the inshore areas of our region. And so the last thing I wanted to mention about data collection is that we do have a number of sort of modern tools and techniques that are becoming more available and more affordable that can help us understand both the turn diets as well as the distribution of fish in the environment. So myself and some colleagues at UNH and Cornell University and some other institutions along the coast have been using a number of these molecular methods to enhance foraging ecology data. So this includes the use of environmental DNA, and we can get really great information by actually sampling the fecal matter or poop of the birds, and we can get much better species-specific resolution of what the birds are eating. So when I was talking about sand lance earlier, we know that there are 2 species of sand lance that can occur in the diets, but they're very difficult to actually distinguish the 2 different species by visual methods. Even if you're holding if you're lucky enough to have 2 of the different species in your hands at the same time, you could probably do the basic meristic or morphology analysis to figure out which fish species you have in your hand. But when you're trying to identify a fish that's in a bird's mouth and it's on the wing and it's moving, it's very difficult to make that definitive identification. Same reason why when I talk about hake, I talk about a hake complex and the fact that we can't quite distinguish between these different species because they're moving and they're very difficult to identify, especially those juvenile and larval life stages. But from eDNA, we can actually get species-specific information when we sequence those samples. And that's important because then we can trace that species-specific information back to thinking about the management of those species and what particular actions we could take to account for those, for the sustainable management of those species to help support the birds. Another technique that we're using is employing an ecological tracer like stable isotope, which allows us to get an integrated signal of the diets of any predator, but of seabirds in particular. And this method can help us gain more insights into life stages of the birds that are difficult to monitor even on these islands and with these great programs that we have. So adult seabirds largely forage at sea, and they're very difficult to observe at sea. So stable isotopes can help give us information on the trophic levels that the birds are feeding at, and give us some information on the habitats that they're feeding in through carbon and nitrogen tracers and in my work, I've been looking at this by looking at the eggshell tissues, hatched eggshell tissues, sorry, of the seabirds, so we can get information on the pre-breeding diets of terns and other seabirds that are nesting on these islands. And all of these techniques give us different pieces of a bigger puzzle, but when they're used together, they're very powerful in giving us a more comprehensive understanding and high resolution view of what's going on in the system. To close, the last thing I wanted to mention is that as we continue to live in an increasingly warmer world, we're going to need to manage seabird, fish, and other fish and wildlife populations under high uncertainty for what we do not quite know yet and what we're still learning about. But we do have a range of tools that can help us conserve and sustain species under highly variable conditions and poor understanding. Some of these strategies include maintaining a diversity of habitats and foraging conditions for the birds so that they have a lot of options for nesting and hunting for food. Another strategy is to reduce the influence of non-climate stressors, such as fishing pressure on their forage populations. And the last strategy is to take a holistic or ecosystem-based approach to fisheries management that accounts for these predator, prey, and other species interactions, and that we leave some forage in the ocean for the birds and other top predators. So with that, I'd like to acknowledge the National Audubon Seabird Institute and the U.S. Fish and Wildlife Service. This work could not be done without them and their dedication to collecting data on these islands and staffing these colonies throughout the chick rearing season. I also wanted to mention that much of the work that I presented on today comes from my former student's master's thesis, Keenan Yakola, who was an island manager with Project Puffin and the National Audubon Seabird Institute before and during his graduate work. Rachel Bratton and Henry Liggett were undergrad and postdocs in my lab and have also made major contributions to the work that I showed today. And my collaborators at the University of Massachusetts Amherst, Adrian Jordan and Chris Sutherland, have helped oversee a lot of this work and helped with some of the modeling as well. And so with that, I just wanted to mention that the Northeast Climate Adaptation Science Center is based at the University of Massachusetts Amherst, and you can learn about this project and much of our other work by visiting our website. And with that, I would happily take any questions that folks might have. So Tina asks, she's interested in learning more about the research on the seabird eggs, and if I could speak more to that. So yeah, this is something that we actually just released a paper on about a week ago. So basically what happens is, as I mentioned, the adult seabirds forage at sea for themselves. And when they actually we have the best understanding of the chick diets because the adult seabirds go out to sea, forage, and bring back the prey to the islands where the observers are able to see the prey being brought back, right? But what they eat at sea, we can't see. It's very difficult to observe seabirds foraging out on the ocean. We do have some observations, but it's nowhere near as comprehensive as what we're getting in the chick diets. And I don't think we're quite there with especially these little terns in terms of putting cameras on their backs to, you see like some of the larger megafauna with like actual cameras strapped to their backs, and you could see them eating things. But just in general, observing organisms foraging at sea is very difficult. So stable isotopes work like ecological tracers. So I think we think of isotopes very often. People are more familiar with like radioisotopes that degrade over time, but stable isotopes are stable, and they exist in all of our tissues. And we can get from nitrogen, we can get information on the trophic level that a predator is feeding at. So we can tell whether it's at the top of the food chain, or if it's at the sort of mid-range of the food chain, or if it's foraging more towards the bottom of the food chain. Whereas the carbon signal gives us information about the type of habitat that the predator is foraging in. So when we have a more what we call negative value for the carbon, we know that that represents a more offshore or pelagic habitat, where when the carbon values are more enriched or positive, that means that the seabird or predator is feeding in more inshore or demersal habitats. So depending on what tissue you analyze, you get a different integrated view of the foraging ecology of that species. So mussel has sort of an integrated history of about a month for most species. Bone has an integrated history of like years. Feathers are often used for the period of a molting season. And eggshells, which is the tissue that I've been focusing on more recently, tells us about what the diet was just prior to the female laying those eggs. So this is a really critical time period to understand the foraging ecology of these highly migratory seabirds, and that some of them have just migrated from Central and South America or the Southern Ocean, and they've depleted their energy reserves to get here. And then what they eat when they arrive in the Gulf of Maine then provides the nutrition and the energy for them to then form and lay the eggs. So the signal that we're picking up from the eggshell is representing what they ate after in about the sort of week to 10 or so day period just prior to laying that egg. And so what we do is we've worked with the island managers who are out there in the colonies, and they're sort of getting ready to monitor the chick diets and the chick rearing period. So they have a really good handle on when those eggs are hatching, and they can link that egg back to an individual nest, and they know what species laid it and so forth and so on. So what we've done is we have this program where we've asked the managers that live on those islands, when those eggs are hatched, and you know what nest they're from, and you know what species they came from, and you know that it wasn't predated by like a seagull or something like that, collect that eggshell, and they put them in these little envelopes, and then they send them back to us at UMass. And then we clean them, and we remove any dirt and debris and whatnot. And then we actually put them in a machine that then tells us what the carbon and nitrogen concentration was in that signal. And then we can compare that information across individuals and species and populations to get a sense of what their foraging ecology was. So the limitation of that stable isotope as an ecological tracer is that it can't tell us what species exactly went into that signal, or exactly where that bird was foraging that produced that signal, which is why if we compare that method with like those visual observations that I was talking largely about today, where we get information on like the species of prey that was delivered to an organism, and then we pair it with this other method, then we can build a bigger picture, because that observation of the prey that was delivered that we see from the bird blinds, that tells us it's like a snapshot in time of what the diet was. But the stable isotope can tell us this more integrated history of the organism over the order of days, months, and years. And I've used this technique quite a bit in fish research. So I've studied large pelagic fishes like tunas and dolphin fish and sharks and bluefish and all sorts of different species. And so that's a little different because we can basically take 2 samples at once from an individual. We can look in their stomach and cut open a fish. We look in their stomach, we see what they ate. We take a muscle sample, and then we have like this automatically paired data set using these 2 different methods. And we've been able to say a lot more about the fish diets because we do more lethal sampling to get their stomachs than we do with the seabirds. But we are starting to use like feathers in the seabirds, so we can then link stable isotopes in the eggs to the feathers of the chicks and the adults. And so there's a lot of different things that you can do. It just takes a lot of coordination and a lot of a lot of forethought to get all those things together. So hopefully that answers your question. And the next question looks like, have increases in butterfish impacted health of puffins and terns? So Steve Kress, who helped found Project Puffin and really established all of these great monitoring methods on the islands, he and colleagues have specifically studied the impacts of butterfish on Atlantic puffin, and we do have more definitive links with butterfish having a negative effect on puffin survival, chick survival, and growth. We are really close to having those results for terns. There's a group in New Brunswick, Canada, that has studied this on Machias Seal Island. So at that particular site, they've made some stronger links between diet and productivity. The data that I've showed you today, we are linking that currently to the productivity of common terns and roseate terns, but that work isn't done yet. But preliminary work is showing that primarily when the birds eat more invertebrates, they tend to have lower productivity compared to their preferred prey. But we have not been able to, we're still working on the modeling to make that definitive link with butterfish. So that's inconclusive at this time.